So I'm just going to do basically a review session for the lab practical. I did do some Zoom sessions with students, and I found out that um, most people just kind of want a little bit of help on the histology. So I'm going to go through this just like I would a review session if I had you guys in person. I'm not going to go through the microscope identification, because you guys probably know how to identify most of the parts of the microscope from 181. So I'll start with the mitosis lab, and I'm just going to go through some of the slides for you. So we'll start with this one here. And so again, this was a fish blastula. So all of these cells are undergoing mitosis, right, to form the larva. More than you probably, again, needed to know, but the marine biologist in me couldn't not say that. Okay, so when you're looking at this slide, first, you're going to see the cells that are in interphase here. The cells that are in interphase, basically, they don't look like anything because you can't see the chromosomes, right? The chromosomes are not condensed because they're all loosey-goosey, right? In the form of chromatin, because they're actually, that you want that DNA available to be used to make protein. So the only time that your DNA, right, is condensing to form these nice pretty chromosomes is during mitosis. So that's where prophase shows up. In prophase, you're going to finally see these nice condensed chromosomes inside the nucleus. And then you'll still see some of that nuclear membrane, even though the nuclear membrane, as you can see here, is really starting to break down. Okay. And then you might even start to see some of the beginning of the spindle formation. Here, this is obviously here is the metaphase plate. So this is metaphase. And then you can see again that mitotic spindle here, and the centrioles would be. Here again, this you can really see is a good prophase. You can still see some of the nuclear membrane, and then you can see the chromosomes condensed inside that nucleus. And then you can see again the centrioles here, where you've got the beginning of the formation of the mitotic spindle and a lot of these astral rays. And again, here you're seeing that same thing here, prophase, all same things, another prophase. Here is metaphase. So again, beautiful metaphase cell there. Here's the metaphase plate. So all the chromosomes are nice and lined up in the metaphase plate. There's your centrioles, the mitotic spindle, your astral rays sticking out there. Again, another close-up of that exact same thing, so you can really see the chromosomes lined up at that metaphase plate. Here is a perfect anaphase, right? So you have those sister chromatids being pulled apart, so to the opposite poles here, towards the centriole here and here. So that's going to be a perfect anaphase. And then here, this is just to kind of test you, showing you prophase again here, another prophase here. So again, just showing you these kind of different cells. And again, most of these other ones here are in interphase. So again, here you got the centrioles. Here the cell is in anaphase. And you can see there's the astral rays. And then, of course, here's the mitotic spindle. And again, you're going to see here now cells that are going to be in telophase or telophase. Telophase is kind of the more commonly used pronunciation, but either, if you grew up with telophase, you can do that, or telophase. So how you know that a cell is in telophase is right here. It has this cleavage furrow. And at the end of this video, I'll have to turn on my camera. I have this great shirt that anatomy students got me from a previous semester, and it has a cell in telophase just like this, the outline of it. And then at the top, it says, "Stop staring at my cleavage." And so again, that's because this is the cleavage furrow. So it's one of my favorite shirts. But again, so this is that cleavage furrow that you can see with your cell in telophase here. Again, close up of this cell in telophase. Again, with the cleavage, this is pointing to the cleavage furrow where it pinches in here. And there's basically like a band of actin filaments that are going to be kind of tightening like a rubber band to pinch this off. And then this is again metaphase, so metaphase plate. Here again, another telophase cell, and you can really see that cleavage furrow forming and almost pinching off these two daughter cells now. And then here's the models. I don't believe that we use the models too much for the purposes of the practical, but this is just showing you again prophase and those chromosomes condensing. Here's metaphase. So, and this is just late prophase, but I don't think we even have you differentiate that. Here's metaphase, and you can see again the spindle. So this is just kind of breaking it down. Here's early anaphase for you. And then here's the beginning of telophase, because you've got the formation of the cleavage furrow where it's pinching in. Late telophase there. And then now you have your two daughter cells. So again, hopefully that helps in terms of just going through some of those slides. So let me go ahead. I'm going to close out of this, and then let's jump right into histology. So let me go back here. I know that histology is usually the most challenging for most students. So let's jump into histology. And I'll give you some hints here on how I kind of teach histology in terms of helping you to identify the different types of cells. So again, this is going to be all epithelial tissue. Now, the tough part is, of course, that you're not going to get all epithelial tissue together, right? In the practical, you're going to have some epithelial tissue mixed in with some connective tissue. And so try to keep that in mind. But the more that you look through these slides and you look through them, especially with a StudyMate flashcards, the easier it should get. So again, simple squamous is pretty easy. I'm going to show you some simple squamous epithelium in some other slides as well, because this is actually just frog skin. But again, you can see these flat pancake cells are the simple squamous cells. Oops. And remember that you need to kind of keep in mind that you should know, I don't even know what I'm doing there. Let me exit out of that. But keep in mind that you do need to know one place. Try to keep one place in mind. I don't know why this isn't. I'm going to go ahead and close this and reopen this to see if I can get it to function a little bit better for me here. So let's try this one more time. Here we go. So again, that simple squamous epithelium, a common place that you can find that is going to be in the alveoli of the lungs. So anywhere where gas exchange is going to have to, occur like that, or just diffusion of substances is going to have to occur, is where you're going to find simple squamous, because again, you need that kind of thin layer. So wow, this really does not want to advance to the next slide. Here we go. So here you're going to see this is actually a cross section of your intestines. So you can actually see some different types of tissue here, which is pretty cool. You're going to see the simple columnar epithelium that is going to line the intestines here. And this is the lumen inside. So again, the lumen is going to be the interior of any type of like hollow organ or, any type of tract that you have. The inside is going to be the lumen. And then if you look actually towards the outside here, you can actually see the smooth muscle. I'll zoom in on this. That lines the smooth muscle lines the GI tract, and then on the very, very outside of the peritoneum, you'll actually be able to see some simple squamous epithelium. So those are kind of the majority of the things. Let's zoom in here and see all the good stuff. So again, these are these simple columnar epithelial cells, really easy to identify because they look like they're just one layer of cells, and you can see all their nuclei are all lined up nice and pretty at the bottom. Again, remember that because this is an epithelial tissue, we can ask you about this. They're called the basement membrane, this structure right here. In which all the cells are attached to. The basement membrane, we can ask you about this. The apical or free surface, either can be used for the practical. We could ask you about these cells called the goblet cells. Kind of always funny, they look like goblets, right? But I love the name, goblet cell. And they secrete, again, the mucus. Here you can see these are actually smooth muscle cells. Which is pretty cool. And these are these kind of longitudinal ones, and then you'll see a cross section of some muscle cells, because as we'll learn, we'll talk about this a little bit later, but you have two layers going in different directions. So here you're seeing a cross section, and here you're seeing a longitudinal section of this smooth muscle. So smooth muscle, and then check this out on the very outside of all of this, you're actually going to see a layer of simple squamous epithelium, these nice flat pancake cells. So this is a cool slide because you can see a lot of different tissues in this one slide. So it's always one of my favorites. Here is a zoom in of some of the smooth muscle here. A longitudinal section, cross section of those smooth muscle cells. And then here, check this out. These are those simple squamous pancake cells, which are pretty cool. Okay. So, and then here's a close-up of those goblet cells. Again, these are those simple columnar epithelial cells. This is actually pointing to the basement membrane or the basal surface. If we ask you what surface is this, you would say the basal surface, or again, the germinal layer. It just depends on how you learned it, but we use usually basal surface because it's up against the basement membrane, so it's nice and easy to remember. And then apical surface or free surface, okay, for here. So this one is a little tough when it's zoomed out, but this is actually going to be, and you'll see in a minute when we zoom in, your cross-section and longitudinal section through your kidney tubules. So this is going to be your simple cuboidal. So let's zoom in. This is a longitudinal section of your simple cuboidal. And so you're basically like, you cut lengthwise on a hose here, and you're looking at the lumen here. So this is the lumen. All of the white areas are the lumen here in this one. And then you can zoom in, and now you can see these simple cuboidal epithelial cells, right? Pretty obvious. And then you can see the lumen on the inside here. This is going to be, if I tag this and say, what surface is this? You would put apical or free surface. If I tagged this back here and said, what surface is this? You would put the basal surface. Or if I said, what structure is back here, you would say the basement membrane. Okay, so hopefully that will help you. So here, this would be what structure, and you would say basement membrane. Okay, or what surface? Basal surface. So again, just this now is a cross section instead of this one's a longitudinal section. So again, these are tubules, kind of like a hose. They're big tubes, and you can cut lengthwise through the tube, or you can do a cross section through the tube. So again, now you're looking into the tube. So here are your simple cuboidal cells. Here is the lumen inside here. And then here is the basement membrane on the outside. This would be your apical surface lining the lumen here. Okay, so hopefully that helps out with that. Same thing again, a zoom in here of this basement membrane. Here's the apical surface lining this lumen here. Okay, apical or free surface. And again, all simple cuboidal. So this one is always a little tough for students. And that's because you always think of stratified squamous epithelium as being kind of in the skin, but this is non-keratinized stratified squamous epithelium. And a student asked me, do you have to put the non-keratinized? No. You can just put stratified squamous epithelium. I'm happy for that. But you don't want to put keratinized on this one because it's not keratinized. So I'll show you the keratinized in just a minute. So this is actually a cross section through a mouse esophagus. And so let's zoom in here. You can see, this is showing you the lumen while you're zoomed out. So you can see the lumen here. And then of course, this is going to be the apical surface. This is going to be the basal surface up against the basement membrane. Okay, so again, basement membrane is going to be back here. So the way that you can tell this is stratified squamous epithelium is that you're going to have, it's going to look like 15 to 20 layers thick. Cell layers thick. So if you're confused between this one and transitional, which a lot of students do, get those two mixed up. The easy way to tell this one is the cells are kind of more flattened and squished together. And in transitional, they're kind of more bubbly cells. And then here you're going to see that you're going to have closer to like 15 to 20 cell layers thick, where in the transitional, it's only going to look like it's about 7 cell layers thick. Okay. So again, here's your lumen. This is going to be the apical surface up against your lumen. Excuse my large dog that's trying to be part of the video here. Sorry, she's on my headphones here. And so excuse me for that. Yeah, so here's your apical surface. Here is the basement membrane back here in the basal surface. Here's the lumen. And again, this is that non-keratinized stratified squamous epithelium, or again, just stratified squamous epithelium. So you can see here, there's no stratum corneum. There's no, there's no keratinized layer up there of a bunch of dead keratinocytes. Okay. So, and again, here's going to be your, oftentimes we call this your germinal layer. You can say basal layer either way. I'm not picky about that. But these are going to be the cells that are going to be your proliferative cells that are undergoing mitosis back here, right up against, again, the basement membrane, like you're going to see in all your epithelial tissue. And then here is going to be your apical surface, and this will be your lumen in here. So here, this is keratinized now, stratified squamous epithelium. And because you can see this keratinized layer here. Now, this is cool because this is a cross section through, of course, skin. So you've done the skin lab as well. So this is kind of cool because you can see from here in the dark purple all the way up here is epidermis. Okay. And this layer from here, actually from about here on up, is all stratum corneum. This is all the keratinized layer. So this is keratinized stratified squamous epithelium. Now this is cool because down here, guys, is dermis. All of this is dermis. And then look, you can see now you're into the hypodermis, or what we call the subcutaneous layer. And you can see there's a lot of adipose tissue, okay? So if we flagged this and said, what connective tissue is this? You would say adipose tissue. And then if you zoomed in on the dermis here, you would actually see that irregular connective tissue in there. So let's go see what we get when we zoom in here. Okay, so again, here is the epidermis, and this is that keratinized now stratified squamous epithelium. Okay, so here is this here is the keratinized layer, and actually this is kind of cool. We'll zoom in on this a little bit later, but you can actually see the stratum corneum up here, and then here's some of the stratum lucidum here, which is pretty cool. So you get stratum lucidum, stratum corneum, but in this slide there, they just want you to say that it's the keratinized layer. Okay, and then of course this arrow is pointing to the fact that this is the apical or free surface. Here, this would be again the germinal layer or basal layer of cells, and then you have them right up against the basement membrane would be the structure there. And then this is all dermis here. And what's cool is, again, you can see, look, this is that dense, irregular connective tissue in the dermis here. So you can see, and I'll talk about the dense irregular connective tissue looking like these kind of woven fibers. So you have these collagen fibers kind of woven going in these different directions. So here again, you're just zooming in on what would be that germinal or basal layer there. And again, that will be right up against that basement membrane. And then here you can see that's that keratinized layer. Here, this should be pretty easy. These cells, again, are going to line places like the trachea. And so this is a cross-section through the trachea. You can actually see some hyaline cartilage here, which is nice. So you can actually kind of test yourself on cartilage as well as epithelial tissue here. So this is the perichondrium. This is your hyaline cartilage. You can see chondrocytes in their lacuna. So again, all kinds of cool stuff. Back here, you're going to see some chondroblasts. Anyway, so you can review that too as you're going through this. But we're here to look at this layer, which is going to be your pseudostratified ciliated columnar epithelium. So let's come here, and we say pseudostratified because remember, this is goofy because it looks like it's maybe like 2 or 3 cell layers thick, but it's not. It's just 1 cell layer thick. It's just that these cells are goofy. Some are long and some are short in here, and then this one comes all the way up here, and then this one might be short in between. So it makes it look like it's stratified, but it's not. It's falsely stratified, hence the name pseudostratified ciliated columnar epithelium. And again, it's ciliated. So that's the nice part. So now you know the pseudostratified, right? And then it's ciliated because it has the cilia lining the lumen. Again, they help to kind of push up the mucus, if you can think this is in the trachea, right? And then of course you have to have these lovely goblet cells, right? That are going to produce the mucus, that will trap all the, particulates. And then of course those cilia push it up to the back of your throat. And then, here, they're just pointing to these pseudostratified ciliated columnar epithelial cells, or if it says what's the structure, it would be the nuclei. So that's basically the bulk of that one. This one here is going to be your transitional epithelium. And so I'll zoom in so you can see this and it'll make a little more sense as to why it's transitional and not stratified squamous epithelium. Because again, those are the two that usually people mix up. So here's the lumen and here's the apical surface. And then I would expect you would need to be zoomed in. So out here it would be really tough to tell. Here it's much easier to tell that you have transitional epithelium now. So now check out how different this looks from the stratified squamous epithelium. You can go back in the video here. Here it's going to look like it's only about 7 to 10 cell layers thick if you count like how many, nuclei, maybe 1, 2, 3, 4, 5. I'd say even, 5 to 7. Again, that's just to show you the vast difference between how many cell layers thick your transitional epithelium is versus, 15 to 20 stratified squamous, epithelial cells all squished together. So that's the easy way to separate these two out. Again, transitional is going to look more like, 5 to 7, maybe 10 layers at the most. And then you're going to see these cells are going to be kind of more, kind of these bigger, kind of more bubbly cells. Okay. Obviously, this is the apical surface here. And here's the lumen. And then, of course, this is the basal surface or germinal layer, if you want to call it. And then there's the basement membrane. Here is really a better picture of transitional epithelium, and you really can see the apical surface is kind of a more bubbly. You have these more bubbly cells, allowing them to stretch. If it was stretched, they'd be flattened. And so again, it looks like it's about 7 cell layers thick there. And then you have the basement membrane. Okay. So hopefully that helps you in terms of differentiating that from the stratified squamous. If you look at them now side by side, they look really different. So I think that was it for all of the epithelial. So moving right along, so my video doesn't get too long here. Let's go down and go to connective tissue. Jumping right along. Connective tissue. Lots of stuff with connective tissue. Okay. So here we go. I'm going to try to go through this as quick as I can. Here is your areolar connective tissue. And so here you can see you're going to have these fibroblasts, nuclei. We're only going to ask you about the fibroblasts. We're not going to ask you about mast cells because it just gets a little more involved than we need to. So any of these nuclei that you see, you're going to see are going to be fibroblast cells, and that's what's secreting, again, the matrix for your connective tissue. So then you can see these little, like really thin threads that run through here. Those are going to be your elastic fibers, and then you're going to see the thicker ones are going to be your collagen fibers. Okay, and then your fibroblast nuclei. Same here again. You'll be able to see your fibroblast nuclei, and then you can see these the thinner purple elastic fibers, and then you'll see the thicker collagen fibers. So here, this is going to be your adipose tissue. It has some blood vessels kind of running through some of it, which you'll see because adipose tissue is vascular, especially in the subcutaneous region. So again, here, let's zoom in. You have your adipocytes. Those are the adipose cells called adipocytes. And you'll see that inside each of them is a lipid droplet. And then what happens is that lipid droplet takes up most of the cell, and then it pushes out the cytoplasm to the very edge. So here you're going to see the adipocyte nuclei back here. And so here's some more adipose cells kind of stuck in here, and they're stuck in between this connective tissue that's called dense regular connective tissue. Now, the easy way to remember dense regular connective tissue is it's going to have a bunch of these collagen fibers that are going to be really packed in tight here. And if you look here, all of the collagen fibers are also running in the same direction here. That's another thing you'll notice about the dense, regular connective tissue. And so it's easy then to remember where you would find this tissue. This tissue is going to be in places like your tendons and your ligaments, where all of the force is going to be pulled in one direction, right? So that's why you're going to have all these tough collagen fibers running right next to each other in one direction, okay? Versus like when we go to look at your dense irregular connective tissue in just a minute, you're going to see those collagen fibers are going to be running in all different directions. And that's because that's going to have to withstand a pulling force in different directions like you would with your skin. But here you just need the pulling force in one direction because this is going to be in your tendons and your ligaments. So here you're going to see this is that dense regular connective tissue with all of these. If we said, what's this structure? These are all the collagen fibers. If we're pointing to these little purple dots, those are the fibroblast nuclei. And again, remember that those fibroblast cells are what actually secreted that collagen. So that's where that collagen came from. Oops. Okay, so here's a close-up of this dense regular connective tissue. You can see here the fibroblast nuclei, and you can see all of those collagen fibers. Again, dense regular connective tissue. This one here is going to be dense irregular connective tissue. And dense irregular connective tissue you find in places like your skin. Like if you pinch up your skin and you pull it in different directions, right? What allows you to do that is this dense irregular connective tissue that makes up your dermis. And so it allows you to pull in different directions because these collagen fibers are kind of woven in these different directions here. And then you can see the fibroblast nuclei here because this is, again, a cross-section through the skin. You can actually see one of the hair roots here. So again, here's another great picture of this dense irregular connective tissue that makes up the dermis. And you can see, again, these collagen fibers, like some of them here are woven this way and then woven this way and then woven this way, all in these different directions to withstand pulling in the different directions. And again, you'll see here's fibroblast nuclei right here. Hard to see in this one, but those are those purple dots. And then here it's pointing to the collagen fibers. So here is looking at that same slide of the trachea. And you've got your pseudostratified ciliated columnar epithelial cells up here. But now down here, you have your connective tissue, your cartilage connective tissue. And then this here is actually the perichondrium. So this here, when we talk about the different types of cartilage, This is hyaline cartilage, and it's going to be this kind of pinkish purple that you'll see in the slides. And you'll look here, back here, you're going to have right up against the perichondrium, you're going to have your osteoblasts. Okay, these are going to be your osteoblast cells that are secreting the matrix. Now, the cells that are farther inside here are going to be your osteocytes. And then this is actually just pointing to that, that matrix, that extracellular matrix that's secreted, again, by those osteoblasts. And if we ask you a question like, name this space, so remember that the chondrocytes, I'm sorry, did I say, I hope I didn't say osteocyte there, but these are chondrocytes. So again, chondra is going to be cartilage. So osteo will be bone. I just did your bone lecture. I just recorded that, so I have osteo on the brain. But for cartilage, it's going to be chondro. So these are chondrocytes, chondroblasts back here that are secreting the matrix, and then your perichondrium. And so again, but just like in bone cells, you're going to have your cells trapped in their lacuna after they've secreted matrix all around themselves. So here is a chondrocyte that is now in a space called a lacuna, okay, with matrix all surrounding all around it. So again, chondroblast, chondrocytes here inside their lacuna, and then perichondrium. Here you're going to see, this is just a close-up of your hyaline cartilage here and your hyaline cartilage cells. Here are going to be your chondroblasts, not osteochondroblasts. And again, you can see them actually undergoing mitotic divisions. Here you're going to see chondrocytes inside the lacuna, okay? So here, this is your elastic cartilage, and you know that it's elastic cartilage because you see these lovely big bubbly cartilage cells, and there's lots of purple fibers. So you know those purple fibers are your elastic fibers. So this is going to be your elastic cartilage. And you find this again in like places like in the external ear. Where you have a lot of elasticity there. So here again, you're going to see are all of these chondrocytes. Again, your cartilage cells. And then here, this purple matrix is going to consist mostly of your elastic fibers. And then, of course, on the outside here is going to be the perichondrium again. So again, a close-up here, you're going to have here these chondrocytes. And then again, your chondroblasts, you can't really see very well, but they're going to be these cells back up against the perichondrium here. And then here, this is just pointing to that matrix. And we would probably say, you know, what fibers would you find here? You would put elastic fibers. Here, this is easy to see. This is a cross section of bone. This is going to be that central canal. This is a bone forming unit called the osteon. So let me zoom in to your osteon here. And inside your osteon, you're going to have your central canal where your blood vessels are running through. Then you're going to have these rings of matrix that we call lamella, or plural, meaning more than one, lamellae. So again, here's a ring of matrix. Here's a ring of matrix. So this is a lamella. And then you'll see these osteocytes. Right? Now we're osteobone. Osteocytes are going to be the cells that are trapped in the lacuna here. And then these osteocytes communicate with each other and pass nutrients through these little tiny canals called canaliculi. So these are all these little extensions are all these canaliculi. So yeah, so this here is pointing to the canaliculi. This is actually pointing to, so here you can see all these central canals, right, in this cross section. And then you see this blood vessel that's running across, this canal with a blood vessel running across the osteons, connecting the central canal of one osteon to the central canal of another osteon. Those are called perforating canals or Volkmann's canals. You can use either term. Volkmann was the person that it's named after. Perforating canal is kind of just the name that makes a little more sense because it perforates through the bone. Again, this is an osteon. These are all these central canals to your bone. This is just a side-by-side of, I'm not sure why it wasn't back with the regular, dense regular, and dense irregular connective tissue, but this is showing you a side-by-side of dense regular connective tissue and dense irregular connective tissue. This is showing you a side-by-side of hyaline cartilage and elastic cartilage. And then this is, again, just a model of an osteon. So you can see the central canal with the blood vessels. You can see these rings of matrix, the lamellae. You can see the lacuna here, the spaces. And then inside would be your osteocytes. And then these little extensions here are the canaliculi. So hopefully that all helps. And so again, the same thing here. Here's your osteons, central canal. Here's your lacuna that are going to have your osteocytes and your rings of matrix. Here, this is nice. They pull up those rings of matrix so you can see them, called, again, the lamella, or lamellae, plural. And then here on the outside, you'll see is the periosteum. So remember, you had the perichondrium before in cartilage. You have the periosteum in bone. So, and this is nice because you can actually see here's these central canals that would be running in the center of the osteons. And then check this out. Here's your perforating canals or your Volkmann canals. So that one's a good one to see that. And I think that is it for the connective tissue slides. So let me go ahead and close that out. We're going to move right along to muscle tissue and nervous tissue isn't. It's a little less involved. So let's go right into your muscle and nervous tissue. Here we go. Lined. Okay, so your muscle tissue is going to start with this smooth muscle. And so this is kind of nice because this is the smooth muscle that lines your GI tract, right? This is the cross-section to the intestines. So you saw the simple columnar epithelium here. So now they're zooming in on the smooth muscle outside that lines the intestinal tract. That's what does your peristaltic contractions. So here is just the longitudinal section through it. Here's a cross-section through that smooth muscle. And then here, this is going to be those simple squamous epithelial cells that actually make up the peritoneum. So again, zoom in on these smooth muscle cells here and here. And then these, you really can see these flat pancake simple squamous epithelial cells. Okay, so here, this is going to be a fascicle of your skeletal muscle. So this is a slide of skeletal muscle. This would be called a fascicle, or kind of a bundle of your muscle cells and skeletal muscle cells. And then let's see, I think you guys do have to know the different parts for these slides. So the easiest way to like kind of look at this is go in layers. So you have to know the different connective tissue layers that bundle everything together. So you're going to do epi is on the outside. Epimysium is going to be the layer of dense connective tissue that's holding this muscle together. And then here, then we have the muscle itself is broken up into these bundles or fascicles of cells. And these are each going to be lined by the perimysium. So here's perimysium, perimysium. And then internally around each of these individual muscle cells or muscle fibers, we're going to see the endomysium. So here is going to be endomysium, here is going to be endomysium. And this is going to be kind of a goofy term, but they call individual cells. So if you're talking about an individual skeletal, and this is for skeletal muscle, individual cells are going to be called myofibers. So this is a single skeletal muscle cell. So it's called a myofiber. This is a myofiber. This is a myofiber. This is a myofiber. And again, this little white stuff that surrounds each one is going to be that endomysium. So then here, this is a bundle of these skeletal muscle cells or bundle of these myofibers. And they're going to be surrounded by this paramycium. And then the whole entire thing would be surrounded by the epimycium. Okay, so again, kind of going, I'm going to go in to out. So the smallest is going to be your myofibers surrounded by the endomycium. Then put a bunch of those together. This makes a fascicle that's surrounded by the paramycium. And then all of these fascicles bundled together are going to be surrounded by the epimycium, which you can't see here, would go on the outside. Okay, so hopefully that helps clear that up a little bit. Again, this is just another cross section of these skeletal muscles, these myofibers. And remember, skeletal muscles are goofy because they have multiple nuclei. So, and they're going to be on the periphery of the skeletal muscle, but they are multinucleate. And then you'll see the connective tissue in between, surrounding each myofiber skeletal muscle cell is going to be the endomycium. Okay. And same here, this is the exact same thing, myofiber here. Again, there's the nucleus and then surrounded by the endomycium here. And then this is just a lengthwise section instead of a cross section here. This is a lengthwise section because your muscle cells are going to be, each individual myofiber is a long cell if it's a long muscle. Some of them run the length of your muscle. So they're these really long cells. So here, this is a myofiber, this is a myofiber. And then of course, this stuff in between is that endomycium that surrounds it. And then you'll see the purple is the nuclei. And again, there's oftentimes many nuclei per a single cell. So here you'll see that here again is a beautiful close-up. You've got the endomycium in between here. This is a single skeletal muscle fiber, or again, a single skeletal muscle or a myofiber. Again, they mean the same thing, skeletal muscle cell, myofiber. And then here's the endomycium in between. Here is pointing to the striations, okay, of the skeletal muscle. So a skeletal muscle has striations, and you can see that quite well here. And again, it's multinucleate. This is going to be cardiac muscle cell tissue. And the way that you know is that you can see the cardiac muscle cells here have striations, but they also have these intercalated discs. Remember that are going to contain those gap junctions, and we haven't talked about it yet, but it'll all make sense again as they pass on. They basically can communicate with each other through those gap junctions and the intercalated discs. So here you're going to have these intercalated discs in your cardiac muscle cells. You're going to have striations. You can see the nuclei in them. But although in your cardiac muscle cells, you'll see that you just have one nuclei per cell. This is a close-up showing you the difference between dense regular connective tissue and smooth muscle. So dense regular connective tissue is really going to show you these collagen fibers. It's going to look more like hair than your smooth muscle. So it's just putting them side by side so you don't get those two mixed up. So again, dense regular connective tissue looks more like hair, and then smooth muscle here is going to look more like these like elongated spindle cells all squished together. And then I don't think these two look anything alike, but this is dense regular connective tissue, and of course then your skeletal muscle. This is cardiac muscle cells and then skeletal muscle. And then here's a really great image of your cardiac muscle cells. So again, you can see these intercalated discs really well. You can see one nuclei per cell. And then this is just the model, of course, showing you the intercalated discs and one nuclei per cardiac muscle cell. And then, of course, on the outside here, you'd have the endomycium surrounding the cells. Here's a smooth muscle cell model, so you can see the spindle. Again, that spindle formation, one nuclei, no striations in this, and then you have some endomycium you can see there. And then your skeletal muscle model, you can see the striations, many nuclei, and then here you can see the endomycium. And I think that might be it for muscle. Moving right along here. And then let's see, you only have one slide for nervous tissue. So let's see. This is the only slide that we would use for, this is the only one that you had to know for nervous. This is it. And so again, you can see this is actually a neuron. And we wouldn't expect you to be able to tell the dendrites from an axon. We haven't even done that yet. So we just want you to know that this is a neuron. And then you're going to have all of these supportive cells. Sorry, I was just double checking because I can't remember sometimes what you have to know for now versus when we go to the nervous system. But you do have to know the neuroglial cells, which are these support cells. So the neuron is going to be the big one. And then the neuroglial cells are going to be these small, these are actually the neuroglial nuclei, the smaller ones back here. So I think that's all you have to know for this is really just that this is a neuron. And then these are the neuroglial cells. I don't think we even get into the neurofibrils for this exam, so don't worry about that. And let's see. So I think that leaves us just with skin. So let's go back. I think we've done all the histology. Yes, we have. So now we're on to skin. All right. So moving right along here, let's jump into skin. And again, I'm just going to go right through the slides. I was just looking for the later lined ones that don't have all the labels, but maybe I don't have them linked here. Images, the skin models. Let's see. I'll just pull up both of these so you can see all of the images of the skin models here. Okay. So here we go. This one, sorry, this one has the labels. I like the one without the labels, but I don't know, for some reason I didn't have it linked there. But anyway, we do have all of the labels here. I kind of showed you this slide before, but this is nice because you've got the dermis down here. If you zoomed in, you'll actually see that denser regular connective tissue here. And then this, from here to here, is all epidermis. Here, all the way up, epidermis, okay? And so here you can see these dermal papilla. So these are the dermal papilla sticking up here, where the dermis sticks up. And I always think of papilla as these like finger-like structures. So dermal papilla are these finger-like structures that are pushing up, the dermis basically pushing up, right? And so here's all your epidermis. You can break your epidermis down into this very bottom layer. The very bottom layer is going to be the stratum basale, which you can't really see very well here. And then you're going to have the stratum spinosum. Then you have the stratum granulosum, which is this little granular layer right up here. And that'll be if you have the stratum lucidum. Remember, you only have the stratum lucidum in the palms of your hands and the soles of your feet. So this one has stratum lucidum here. And then you have the stratum corneum here. Okay, so this is what you saw when you looked at that keratinized stratified squamous epithelium. All of this is that keratinized stratified squamous epithelium. So just to put it all together, because there's so many terms, your skin, okay, consists of the dermis and the epidermis, okay? The epidermis consists of keratinized stratified squamous epithelium. The dermis is going to consist mostly of dense irregular connective tissue. Some areolar connective tissue will be in the dermal papilla. Okay, so hopefully that puts it all together. Then what we do is we break down the epidermis into different layers. So all of this is still keratinized stratified squamous epithelium as its tissue type. But then we break this tissue up into the different layers for the epidermis. So again, the layer that's doing all the, mitotic cell divisions, that's the stratum basale, okay? Or also called the germinal layer because it's making the next, next generation of cells. And then here's stratum spinosum right above it. Then stratum granulosum is that granular layer. We'll zoom in. Lucidum is here, and then corneum. Okay, oops. So hopefully that helps. Again, this is just a zoomed in portion, so you can really see these dermal papilla here. And now let's just talk epidermis. So again, layers of the epidermis, stratum basal, okay, or that germinal layer, whichever one you want. Then we have the stratum spinosum here. It's going to be the next layer up. Then here you can see, I love this name because it actually matches what it looks like. So this is the stratum granulosum, and you can see it looks kind of darker purple because you can see those little granules in it, right? So there's your stratum granulosum. Here's stratum lucidum. So again, because it's lucid, another nice name, right? And then the stratum corneum is going to be right above that. Excuse me. So this is just a zoom in of the stratum corneum. And this is a zoom in so you can see that stratum corneum and that stratum lucidum. There's the stratum granulosum and then spinosum down below. So again, this is kind of nice because it actually, the names actually make sense here. So again, you can see all the layers here, all 5 layers, stratum basal, stratum spinosum right above that, stratum granulosum, really obvious there. Here's stratum lucidum, very obvious here because it's lucid, and then stratum corneum. And then you can see these lovely dermal papilla here sticking up again from the dermis. So here is just human scalp. So again, you're looking a little farther down in the dermis. Now, if you were to zoom into this tissue, you'd actually see the dermal papilla consists mostly of areolar connective tissue. This one the slide isn't good enough for you to see that. But if you want to remember, if that helps you remember a place where you find areolar connective tissue. And then now below the dermal papilla, in the reticular layer of the dermis, which is here on down, you're going to find that dense irregular connective tissue. So here, this is all that dense irregular connective tissue. And then this is just zooming in on human hair here. So you can see this is actually going to be the root of the hair inside. So this is the root of the hair. We call it the hair root when it's actually underneath the surface. And then here you can see this is the hair follicle, the follicle tissue surrounding it. And then here you can see this is all sebaceous gland. And then here's, so sebaceous gland is always going to surround. If you have a gland that is dumping into the shaft of the hair follicle, then you know it's sebaceous gland, okay? And then you can see also the muscle, if you see muscle that's associated with that hair, that's going to be your rector pili muscle that creates your goosebumps. And then over here, not associated with the hair at all, is going to be your sweat gland or your sudoriferous gland. And you'll see it'll look like just a bunch of tubules. And then usually we'll give you a picture of one that has a nice duct going all the way to the surface. So this is more sebaceous gland associated with a hair follicle. Oh, and there is one error on this slide. Technically, the hair shaft is what we call the hair above the surface. And then below the surface, we actually call it the hair root. So technically, this is the hair root. And so let me see, let's get on to the next slide. So again, this is just zooming in on this hair follicle, and you can see a little bit of the hair root there. And then this is that sebaceous gland. The nice part about sebaceous gland too, when you zoom in, is you can see it's going to look white because there's going to be a lot of oil in there. Think about your sebaceous glands again, are oil glands. So they're going to look, the cells almost look a little bit like your adipocytes because there's so much oil in them. So here's your sebaceous glands, again, dumping into that hair follicle. Here's the erector pili muscle. So you'll see some of that when you look at some of that muscle tissue. It's really the only muscle you're going to be seeing in these slides. So that's kind of the easy way to identify your erector pili muscle. Here is going to be a sebaceous gland here, or a sweat gland. And so it's just going to look like you cut through a bunch of these tubules. Here's another, this is pretty sebaceous gland. Again, lots of oil you can see, so it's going to look white in there. Here's a cross section through a hair follicle. And then there's the hair root inside. Again, sorry, it says shaft. It's technically the hair root because shaft would be at the surface again. And then here, this is cool because you can see your sudoriferous gland, your sweat gland, and then look, the duct, it's kind of broken a little bit in the slide, but you can see where it would run. So here's your duct all the way, that goes all the way up to the surface. Okay. Here, your Meissner's corpuscles, again, are one of your touch receptors in your dermis, in your skin, and they're going to be in these dermal papilla. Okay, so here, remember, this is all epidermis. And then here, these are the dermal papilla, and that's where you're going to find your Meissner's corpuscle. So again, dermal papilla, Meissner's corpuscle. You also, down deeper in the dermis, will find your Pacinian corpuscles. The cross-section through them looks like a cross-section through an onion. So that's an easy way to remember them. And usually they're surrounded because they're deeper. Oftentimes at the kind of basically in between the dermis and the hypodermis, and that transition area, that's where you're usually going to find them. And then there's a sweat gland, and they're mixed in there. I don't know why they put those, but I guess some students might get them mixed up maybe, but I don't think they look anything alike, the Pacinian corpuscle versus an osteon. Here's a skin model. So we will probably use the skin models on the exam because they're nice and clean. So what you can see is you can break it down into, here's the epidermis, right? Here's the dermis, and then here's going to be the hypodermis down here, okay? So, and the hypodermis is going to have all of that subcutaneous fat, so all that adipose tissue. And you can see the Pacinian corpuscle is down even into the hypodermis sometimes. So it's usually right at the border of the dermis and hypodermis. So there's one of your Pacinian corpuscles, one of your touch receptors. On the opposite end here, you can look, remember that when we talk about the dermis is actually broken into two different layers. And actually, let me go back. I want to start with the epidermis and then go into the dermis. So keep in mind that this whole thing is the epidermis, and then it's broken up into its different layers. So this one is not on the soles of the hands or the feet, so it doesn't have the stratum lucidum. So going from top down, you have the stratum corneum, then you have the stratum granulosum. This is stratum spinosum. That very bottom layer, which you really couldn't tag here, would be the stratum basal, okay? And so then now, that's all the epidermis. Now you get into the dermis, right? From about here on up is the dermis. And remember, you can break the dermis into the papillary layer right here, and then the reticular layer. The papillary layer is typically going to have areolar connective tissue, and then the reticular layer is going to have dense irregular connective tissue. You can see in this papillary layers where you're going to find your Meissner's corpuscles. And then let's talk about, and then the reticular layer of the dermis, you're going to find all kinds of things. So this is where you're going to find your hair follicles running with your hair root inside. You'll see the sebaceous gland associated with that hair. Here's your erector pili muscle associated with that hair that can make your hair stand up for your goosebumps. At the bottom here, this is something that you really couldn't see on the slides. You're going to see this little finger-like structure sticking up into the hair follicle, and that's going to have all the blood vessels and nerves associated with it. And so that's called the papilla of hair, which is so nice and easy because, again, it's a papilla, finger-like structure of the hair, okay? And so it'll be sticking up in here. Hopefully that kind of helps you remember some of those structures. Let's see if there's anything I forgot. Again, the Pacinian corpuscle. And then, of course, down here is the dermis. Oh, I forgot. Oh, of course, beautiful sudoriferous gland here. So this is your sudoriferous or sweat glands. I don't care which one you use. And then here is going to be the duct, the sweat gland duct, or the sudoriferous gland duct that goes all the way to the surface. Okay. I think we caught them all in there. So you can see all of those same things on this. I like the papilla of the hair. You can see that a little bit better here. The Pacinian corpuscle looks a little bit different on here, so I like to point that out, but everything else is pretty similar. Sebaceous gland here. Again, your hair follicle with the hair root above the surface is where it's technically called the hair shaft. So again, all the same things here, pretty easy to see in terms of same types of structures. I think that is it for, that would be our last slide here. I do think there is another, I do have another, a bunch of skin models here. No, that's the same group of slides. I thought we had some different skin models, some extra skin models. But I think actually those showed all of the ones that we had. So hopefully, again, that gives you at least a good review of kind of what to see on a bunch of the slides. And I squished it all into 1 hour review session. So if you have questions on these, please let me know. But hopefully this video will help you out a little bit in terms of just giving you a quick down and dirty review of basically most of the material for the lab practical. Now, please keep in mind for the lab practical, again, spelling does count, but keep in mind that, you know, if you mix up a letter, and it doesn't really make a difference in terms of the meaning of the word, I'm not going to take off for that. If you mixed up like 2 to 3 letters and it wasn't, you know, again, it didn't mess up the meaning of the word, I might take off half a point to a point. It just depends on, again, what it does to the meaning of the word. So try to keep that in mind. Now, if you really botched it and I can't even tell what it means, then I have to take the 2 points off. But so again, if you have questions, please reach out to me, and hopefully this review session will help you out just a little bit.